What's important here is that we can sustain this editing effect with both delivery methods for several weeks. So, okay, so it seems like, yeah, it's possible, right? You can most likely permanently cure monogenic diseases, but how safe is this approach? So when you compare AAVs with LNPs, with the AAVs we saw that we actually have the expression. So here we co-express RFP with the base editor, and we have the expression over several weeks. We basically don't lose the base editor. The base editor keeps being expressed over several weeks. When you look at the LNPs, the story is very different. You basically have just a period of time where the base editor is expressed, and then after 72 hours, we cannot detect it anymore. However, the editing is there. So you just need a very short period of time for the edit to occur, and then the base editor is gone again. And additionally, what we saw is when you look at the adaptive immune response, we actually saw antibodies to the base editor when we used AAVs, but we did not see any antibody response when you used LNPs in mice. Then we further looked into safety. So there was this concern that base editors might induce RNA off-targets, so they would randomly deaminate adenines on RNAs. And we actually saw that in vitro as well. So if you look at the first two bars or areas here, you see the in vitro analysis, and we saw that in vitro we actually see a lot of unspecific adenine edits that were not intended. But when you look in vivo, we actually see like far less, and also with LNP, we just see it for a short period of time, and it's gone after a week for sure. And we explain this because the dose is far lower. So in vitro, we have like almost like 80% of the transcripts are actually our base editor. So obviously, you would expect off targets if you highly overexpress such a base editor. But if you look in vivo, our expression levels are far lower, and this probably also explains why we see less off targets. But nevertheless, you might say, okay, RNA-seq is very noisy. There's a lot of transcripts. We might not see everything. So the more important question is actually what happens on the DNA level. So what we did here is we took the primary hepatocytes from the mice, which were edited, and then we clonally expanded a single hepatocytes. We did this because if you think about bulk sequencing and there would be an off-target that is very rare, you would not find it in the noise of all the other DNA from other cells. And if you would do single cell sequencing, you also have a high error rate by the method itself, so you would also not find it. So we thought we just exploit nature itself, and we take the cell, let it expand, make accurate copies of its DNA, and then do whole genome sequencing. And what you then get is a so-called 96 nucleotide graph from each individual clone, where you see the different conversion rates within the whole genome and the DNA that we find. And if you look at the fifth column here, you see the T to C conversions, which is also including the A to G conversions, which are the conversions that should be done or should be found when you use adenine base editor. And when we now compare this to control clones, AAV clones, and LNP clones, we actually did not see any increase of these edits. So we could not find any edits on a DNA level. And then to even further prove the safety, we came up with a long-time study in a sensitized mouse model. So we had mice that had a knockout of P53, so the major tumor suppressor gene, in the hepatocytes, and we confirmed this, that the P53 is really gone. We injected them with the base editor, so we made sure that it also worked, the base editor was in there by checking the PCSK9 levels. And we also checked the editing in the end. And they really had, for over a year, they had base editor in. And then we looked at the livers, and we even gave the samples to a pathologist, and we really did not find any signs of any cancer formation. So this long-term study even further seemed to show that there cannot be a lot of off-targets happening that would be very detrimental. Okay, so we think the approach might be pretty safe. So now the next question was, can we translate our findings? So would it be feasible for clinics? And for this, we actually were able, so I have to thank here already Acuitas Therapeutics, because without them, it would not have been possible to do this big non-human primate study. And here we tested with the LNPs whether or not we can have efficient editing in a large animal model. And what we saw is that we actually achieve it. So using 1.5 mg per kg, we actually have editing rates of around 30%. So these editing rates are high enough to significantly lower PCSK9 and also LDL levels. And then we further looked into the innate immune response and also liver toxicity. And what we saw is like right after injection, so if you look at the gray and the dark yellow bar, these animals got a second dose. And every time after injection, you got like a very quick increase, but it goes back to baseline very quickly. So this again speaks for the LNP approach because it is so transient that you just need it for a short period of time, but then after editing it happened, you don't see any side effects anymore. We also here looked at adaptive immune response, and interestingly, actually here with the LNP approach in comparison to the mouse approach, we saw adaptive immune response to the base editor. So this underlines furthermore that a large animal experiment is important because the immune response in large animals is probably more sensitive in comparison to rodents. So yeah, so altogether I think I could show you that it probably is possible to permanently cure monogenic diseases. I would think it's a pretty safe approach in comparison to other approaches out there yet. And I think it's also possible to scale it up, and theoretically it would be feasible for the clinics. And with this, I would like to already acknowledge the people that were helping. So I would like to thank Melissa, Paulo, Ying, and Sean for the help there from Macaques Therapeutics and made this big study possible. And also would like to thank the people from Syntago that helped with the production and also the large and also the small scale experience.